Hello, everyone. Thank you for joining me today. My name is Uri Bertocchi. I'm a neuroscience PhD candidate from Tel Aviv University. And today I'm going to share with you some preliminary results from my research project, where I studied the epigenetic mechanism of psilocybin therapy for depression. So first, I'm going to give a quick overview on depression, the current treatments, and psychedelic therapy. And then I'm going to dive into the study. So major depressive disorder is a prevalent disorder that affects around 300 million people around the world, and it's a leading cause of disability. The depressed patients often suffer from psychological symptoms like low mood and cognitive difficulties, but also from physical symptoms like weight loss, appetite loss, and fatigue. Around 30% of the patients do not react to any kind of therapy, and they are diagnosed with what we call treatment-resistant depression. And the current treatments that we have today are mainly a combination of medications and psychotherapy, where the most prescribed medications are from the family of selective serotonin receptor inhibitors that help around 60 to 70% of the patients, but leaving many, as I said, without any kind of relief. These medications also have some downsides. First, it takes around 4 to 8 weeks to achieve their fully therapeutic potential. But many patients need to go through a period of trial and error in order to find the right medication for them. Many patients also suffer from residual symptoms and unwanted side effects like sexual dysfunction, sleep problems, and also, when patients want to discontinue the medication, they suffer from withdrawal symptoms. So this leaves many patients with an unmet need for more effective, long-lasting treatments than what we have today. In the last few years, there has been what we call a psychedelic renaissance, where there has been an increased interest in psychedelic compounds like psilocybin, which is the compound that I'm focusing on in my research. As you can see here, there has been an increase in the last few years in publication mentioning psychedelic therapy for psychiatric disorders. And as you can see in this landmark study done in Imperial College London, one dose of psilocybin can effectively reduce depressive symptoms in depressed patients from 1 week up to a few months. And a follow-up study saw the same effect 6 months after the one dose of psilocybin. So this highlights the potential of psilocybin for the therapy for depression. So as I said, psilocybin was vastly researched in the past few years. We know it operates through a unique mechanism where psilocybin binds to serotonin receptors in the cortex, and this creates a wave of neural activity. And this is what we call the psychedelic trip, as you can see here in the animation. But during the trip, and even months after the trip, there's a phenomenon we call in neuroscience a neuroplasticity window, where neural changes in the brain are being set. And this is what we believe that causes the lasting effect of psilocybin on depression. So one of the questions that remain is how is that we see neuroplasticity or neural changes in the brain long after psilocybin has cleared the system? Because in SSRIs, as I said earlier, you need to take the medication every day in order to receive the therapeutic effect. So our hypothesis is that psilocybin activates the serotonin receptors in the cortex, which create a cascade of events that ends up with epigenetic remodeling of the brain cells that drives the prolonged neuroplasticity that we see in the brain that causes the sustained antidepressant effect. So in order to test this hypothesis, we conducted a pilot study just to see whether psilocybin has any epigenetic effect on the brain. We took 2 groups of mice and we injected them intravenously with either psilocybin or saline. And 72 hours after that, we harvested their medial prefrontal cortex, which is a region that is connected to mood
regulation, and dysregulation of this region often connected to depression. So we took these samples and we did nanopore sequencing on them, which enables us to detect both 5-methylpsilocybin and 5-methylhydroxypsilocybin levels in one run. These epigenetic markers are very stable and very abundant in the brain. So we looked for differentially methylated or hydroxymethylated regions between the two groups. We defined the region as 2 to 2,000 similarly methylated CPG sites, and we defined a differentially methylated region as at least 30% difference between the two groups. So when we looked for differentially methylated regions, we found 51 regions. Then we did gene ontology analysis, which is the process of grouping together genes in order to find a shared biological process for these genes. So as you can see in the graph, the more the right the dot is, the more genes are linked to this biological process. And we found an enrichment for 4 biological processes that are not very clearly related to neuroplasticity or brain processes. But then, and this is the interesting part of our results, when we looked under the same rules for differentially hydroxymethylated regions, we found 1,400 of these regions. Gene ontology revealed that these regions were highly enriched for biological processes that are very related to neuroplasticity, like synapse assembly, synapse organization, and neurodevelopment. Also, when we looked for an overall pathway for these regions, we found an enrichment for glutamatergic synapse, which dysregulation of this pathway is known to be connected to various kinds of psychiatric disorders and depression. So this highlights the potential of looking into hydroxymethylpsilocybin as an epigenetic marker that might drive the neuroplasticity in the brain long after psilocybin has cleared the system. But when we try to think about why we find so many differentially hydroxymethylated regions in comparison to differentially methylated regions, first, we know that hydroxymethylation is very abundant in the brain, especially in comparison to other tissues in the body. But also we know that that enzyme that oxidizes methylpsilocybin into hydroxymethylpsilocybin operates much faster than DNA methyltransferases that methylate psilocybin. So this might explain why only after 72 hours we find many changes in hydroxymethylation in the brain and also many changes in methylation. But this leads me to the current state of the project, which we're expanding the pilot study. First, we want to replicate the results using a larger sample size, but we're also including more groups. Some of them with depressive-like symptoms in animal models of depression, but also more time points. We want to try to see whether there are any epigenetic changes for a longer period of time, but also if there's any changes between the dynamics of hydroxymethylation and DNA methylation. And lastly, we're adding RNA sequencing data in order to link the epigenetic changes that we found to gene expression. And hopefully this will open up new avenues for drug development and therapy for depression. So lastly, I want to thank my PI, Professor Val Evenstein, my lab colleagues and our collaborators at the Hebrew University of Jerusalem that did the animal work in this study. And thank you for listening. I hope you find this presentation interesting. And I'm looking forward to answering your question.